Sure.
Good morning, everybody. I welcome you all to our annual Carrot Research Symposium today, and I'm Jaspreet Sidhu, Vegetable Crops Advisor, Kern County. Before we begin with our presentations today, I have Julia here from ANR Program Support. She would go over the housekeeping rules and also share some information on the DPR credits. Julia.
Great, thanks, Jaspreet. So welcome, everyone. So just briefly going over how we can participate today. We are in a Zoom webinar, and so if you have any questions for the speakers, you won't be able to unmute yourself. So you'll want to type those questions into the Q&A that's on the ribbon bar at the bottom of your screen, and we will moderate those questions to the speakers as time allows at the end of every presentation. If you have any comments or just discussion or any technical issues that you're having, please put those in the chat, but leave the questions for the speakers to the Q&A box. And then if you want everyone to be able to see your chat, you'll have to change that little drop down to select all panelists and attendees. And then moving on to the important stuff for today. So if you need DPR credits for this course, continuing education, we do have 1.5 hours of other approved for today's meeting. So here are the requirements. You have to be logged into your computer so we can confirm your attendance. If your name does not appear in your little name box, please make sure that you rename yourself and include your full name first and last so that we can track your attendance and know who you are. You'll have to be present for the entire duration of today's meeting. Only one user per device can receive credit. So if you're sitting there with another person by your side, make sure that they're calling in separately or logging in on another device so we can track both of your credits. You'll need to take the polls that happen at the end of each talk that qualifies. Most of the speakers today do have DPR hours that qualify, so most of them, there'll be 1 or 2 poll questions at the end of their talk, so make sure you participate in those. And then you'll need to take and pass a final exam that will be emailed to you by 5 p.m. today. You'll have to pass with a score of 70% or better. You'll receive that email from ANR Program Support at ucanr.edu. And those questions come directly from the poll questions. So make sure you're paying attention to those poll questions and you'll be able to pass that exam, no problems. So with that, Jaspreet, I will turn it back to you to kick us off for the day.
Thank you, Julia.
Sure.
So we have a pretty packed agenda today, and we will be covering different topics. So without any further delays, I will begin with my presentation, providing updates on the applied research projects from Kern County. Alrighty, so I will be providing updates on the Kern County trials. So in 2022, we did several trials. We did variety trials, which was our organic trial and conventional trial. We did cavity spot fungicide screening, nematicide screening, and then the organic fungicide screening for Alternaria leaf blight. We also have another trial going on, which is the herbicide trial. I haven't mentioned it here. I will be presenting it at the next meeting. So first, the organic variety trial, which was planted in the fall on August 8, 2022, and it was harvested this year on January 18, 2023. We had 61 entries in this trial from Organic Seed Alliance Oregon, and also additional lines from USDA's breeding program run by Dr. Phil Simon. And for the data, we took the assessments for top height, foliar disease severity, root weights, root shape, uniformity, and smoothness. I have provided that data to Dr. Phil Simon, and I will also be sharing this data in the annual report. And in this trial, we found several interesting and several good-looking varieties like this R245 and the Reds 21. And also one of the interesting varieties in this one was this purple, yellow-purple dot population, which was like very interesting, looking like an eyeball in the purple carrot. The conventional variety trial, it was planted just a week ago, and we had 38 cut and peel, 34 cellos, and 30 colored varieties in this trial, and these varieties are from 5 seed companies and from Dr. Phil Simon's USDA breeding program. Next trial is our cavity spot fungicide screening. In this trial, we had 4 organic fungicides in addition to 2 conventional, Ridomil and Reason, and the untreated control. This trial is still ongoing, and we will be harvesting this trial in the next 3 to 4 weeks. But just to give you an idea, like we are seeing good disease pressure in this trial, and we just took some random samples from the buffer rows, and we saw these really good looking lesions on the carrier. So we are pretty sure like we have a good disease pressure, and we will have some interesting data from this trial. Our next one was the nematicide screening trial. We had 6 products in this trial. We had 2 different rates of nimids. Nimids low rate, 5 pints per acre, and nimids high rate, 7 pints per acre. We had an organic product, MB1, and then we had the product Salibro from Corteva and the developmental product from Syngenta. Nimids was applied both the treatments 14 days before planting, and Salibro had 2 applications, one at planting and then one 28 days after planting. The developmental product was applied only at planting, and for the organic product, it was at planting followed by applications every 30 days. And this is the scale that we used for rating the carrots for galling on a scale of 0 to 10. 0 means completely clean roots without any galls, and 10 means like totally galled roots or extensive galling on the roots. So we observed moderate to severe levels of galling in this trial. And another thing that we observed in this trial was significant levels of phytotoxicity, poor seed germ, and loss of plant stand in the higher rates of nimids, which was 7 pints per acre. For the root galling data, we found that the developmental product from Syngenta and the Salibro from Corteva, they had significantly lower root galling compared to the control as well as nimids high rate. While the 2 products, the organic product MB1 and the nimids, a lower rate 5 pints per acre, they performed moderately in terms of root galling in this trial. And likewise, the data on the percent marketable carrots, we had significantly higher marketable carrots in the developmental product from Syngenta and Salibro. Nimids and the organic products were intermediate, while we had the lowest marketable carrots in nimids, higher rate, and the control, untreated control. I have also mentioned in the past as well that nimids has another potential benefit. It performed really well against nut sedge, as you can see in these plots marked with the red flags. Those are the plots that were applied with that. We applied nimids on those plots, and you can see like completely clean or almost clean beds without any nut sedge in these plots. So that's an additional benefit to nimids. To summarize this trial, Salibro and developmental product appeared to provide good protection against root knot in these trials, but we do not know the registration status, like how far we are from registration. Hopefully soon for Salibro and developmental product in couple years. The next nimid is registered on carrots in California, but it still has some phytotoxicity issues, which we will be trying to explore more, like how we can fix those. It only has a caution label, and it has no reentry interval. It's a really great product. Only thing is we have to figure out how to apply it to reduce the phytotoxicity. The organic product also showed good potential in this trial, but further trialing is needed for this product. And we will be doing it this year in 2023 trial. The next trial was our organic fungicide evaluation for alternaria leaf blight. We had 6 products in this trial. We have 5 of these organic products, and one of the product has 2 different treatments, one without any adjuvants and one with an adjuvant. And then we have EcoSwing, we have Cueva, and Howler. There were 6 applications of these products at different intervals in this trial. So this is the data on percent disease incidence. At the beginning, the percent disease incidence varied between 0 to 5% in different treatments, and it increased gradually over time. And by the first week of August, the disease incidence varied between 25 to 45%. Although the disease incidence was not significant, statistically significant different among different treatments, but the treatments Howler and EcoSwing had lower, numerically lower disease incidence compared to the control, and they had about 25%, while the control was 45% of the foliage damaged. And here is the data on the yield. The bars show the data on yield per plot, and the line on the top shows yield per acre. As you can see, there were no statistically significant differences among treatments, but the treatments EcoSwing and Howler had higher yields compared to the controls. And when we extrapolated it to the yields per acre, they still had higher yields, and though they were not significantly different, but when you look at the yields per acre, it would mean like higher returns in the range of $500 to $1,200 per acre in these two treatments compared to the control. So this was our organic trial, which we will be repeating with more organic products this year, and some recommendations from the carrot industry. So moving forward this year, we will maintain the cavity spot nursery and continue screening biologicals, as well as some breeding lines from Dr. Simon's program. We will establish a new root knot nematode nursery to increase our trial area and do large-scale trials, and also to expand our variety screening for root knot nematodes. And we will also evaluate a combination of nematicides and soil surfactants as pre and post applications. And we will continue evaluating the organic and conventional fungicides for alternaria leaf blight management. And we will continue with our conventional and organic variety trials. With this, I would like to acknowledge Dr. Isolde Francis from Cal State University, Bakersfield, for providing us and the inoculum for our cavity spot nursery, and also my team, Jed DeVos, Jennifer Funberg, and my student intern, Crystal Hernandez. And I would like to acknowledge funding from California Fresh Care Advisory Board, help and guidance from Bolthouse Farms, Green Bay Farms, and also funding from Bayer and Go On USA. With this, I would like to say thank you, and we will be open for the poll questions. So here are the poll questions. The first one is, which of these nematicides are registered for use in carrots in California?
You can read the second one too.
Second question, alternaria leaf blight is caused by which pathogen?
All right, great. So we'll give everyone about a minute to answer the questions. In the meantime, Jaspreet, there was a question in the Q&A.
Okay.
If you want me to read that to you.
I got it.
Or you have it, okay.
Okay, so the question is like, when were the nematode trials planted? So we usually plant it mid to late June, so that we have enough population and of the nematodes, and they are active when the soil temperatures are high.
There's one more that came in.
Yeah, so it's the rate for Zonix, and I think I will get it to you. It was not per acre rate. They just told us per plot rate for the active ingredient, and I will share it with you. So for the first one is correct. 65% of people got it correct. It's Nimitz. And then the alternaria leaf blight is caused by Alternaria dulce, which 44% people got right. So.
Awesome.
That's it. And with that, I would like to say thank you. And we will move on to our second presentation.
Hello all, today's talk is evaluation on fungicide performance delivered by solid set overhead sprinkler system on ordinary leaf blight, and some work that we are planning to do on cottony rot. We have 4 people, including myself here. Bob Hockmuth is a regional specialized agent on vegetables working with me closely. We also have Derek Hurley, who is the lab manager for this particular project, who is here with me in this room. And Susana De Silva is also a key person involved in this project. So I just want to acknowledge all of them to begin with. So just to give a perspective, which you all know, but the fresh and processing market carrot production value is around $831 million as of 2021. In North Florida, the planting dates can range from August to March. The common cultivars are listed here. Some of the major issues for us here include alternaria leaf blight, cottony rot by Sclerotinia sclerotiorum. Root knot can be an issue for us, and we've been seeing a little bit of southern blight in the recent years. When we think about alternaria leaf blight, for the right conditions are warm, wet conditions that are ideal for the incidence and further spread. It begins as small water-soaked lesions on the leaf and develops into small black or brown lesions with yellow margins, resulting in complete necrosis at really advanced stages of the disease. They often are seen on leaf margins, can be present on the stems, and they can really move fast within its system under perfect ideal conditions. When we think about the foliar damage, one of the important things when we think about with typical carrot harvesters, at the top pulling part, you're looking at above 50% of the leaf canopy to be available at the end of the studies or end of production so that it can be easily harvested. And ordinary leaf blight can lead to severe defoliation, which can affect this harvesting approach and can decrease yield directly. There are two major pathogens that we have found in Florida associated with the disease. One is the Alternaria dorsii and Alternaria alternata. Alternata is, of course, a much more milder pathogen in that perspective, but we do find a lot of alternata also involved in addition to dorsii. When we think about disease management strategies, we're thinking about clean certified carrot seed, but of course, coated seeds are a limitation in pertaining to organic seed production. Resistant varieties, if available within the scope of the regions and the areas, that is also a possibility. Fungicide use becomes a critical aspect to control the disease, and rotation programs are important to avoid resistance issues. And crop rotation continues to be an important aspect for consideration to reduce inoculum levels. I just want to show you an example of a large-scale field trial that included predominantly conventional fungicides and a few OMRI labeled materials. I'm not going to detail of which materials, but just want to show you all by the end of spring, around May, late May, what we would typically see on a study is we would have plots with very high incidence with really dark sections, which indicates to 50% or way more defoliation, and some of those plots you can see way more, while the effective treatments you can see with much more greener foliage, and you can see that trend throughout this study site right here. Now, this is another study just to give you an example of what we plan to do and what we expect to achieve. This was a study done in spring 2021 with only OMRI listed materials. And as you can see, a significant number of treatment plots are essentially burned down completely because of Alternaria leaf blight, compared to plots that you would see that still holding up. So there are some materials out there that could still hold up well within the OMRI listed platform that could be useful in production system. The rationale of our current study is that what we look at a lot of the times when we are making recommendations to producers, we are looking at data that are generated by backpack sprayer system pressurized by CO2. However, when we look at commercial system in California and many other states, a solid set overhead sprinkler system is used for fungicide delivery. And one of our running hypotheses in this particular case is that we want to look to see whether fungicide performance delivered by solid set irrigation system, could it vary in comparison to the experimental trial data approaches that we typically use and for recommendation to growers. Our experimental setup included plot dimensions of 40 feet by 24 feet. We have 4 beds per plot. We have each of the single bed have 8 rows of carrots. We have 16 treatment plots, which include 4 replicates per treatment. We are planning to do tentatively 6 chemicals in the structure with 2 checks, water controls, 2 application methods. One is tractor spray and the second is a rices irrigation injection approach. Just to give you a perspective of the plot layout here, you're looking at just one treatment plot here. There are 4 beds right here with 4 rices. And if you look at the rices, the way the system is set up is that there's about 180-degree spray angle for the last 2 rices and a 360 for the middle 2 to give full coverage of the entire 4 rows out there for each treatment setup. And if you were to run the full irrigation through the full unit, we should have it fully covered, and the system required a lot of calibration to get it to this stage, and thanks to a great team of people working here. But this is where we are with the irrigation system with the rices, so that all the beds can be irrigated and fungicides delivered within dependent plots as needed. After a particular irrigation event, this is a drone footage taken by Derek Hurley here. And what you're seeing here is a good coverage of the entire study site by that irrigation approach. Each of the red boxes represent one treatment plot. And of course, there are 4 replications for each of them, and they're randomized within the structure. We have Telone 2 used as a fumigant. We had pre-plant fertilizers going at a 15/020 on January 17th. And we also had the variety used is Maverick, which was seeded on the 18th with the seeding rate as shown here. Tentatively, we still need to work this out fine. And we hope to get further inputs from producers pertaining to this. But however, this tentative treatment list is based upon many years of study from 15 to 22, and the findings from those studies are indicating to us that a combination of this set of fungicides could be useful to test in the structure. So we have Stargus. This is a newer material that we are testing for Alternaria, and we've seen some interesting results, so we want to continue that. Also have done pretty well against Alternaria leaf blight in studies here, so we want to continue with that. Nordox would serve also as a control within the study, and then we have Miravis Prime, Merivorn, Luna Sensation, and Fontalis. Now, we are not finalized on this. We still are gathering inputs that would help us to finalize this structure. But this is the same set of treatments that go into tractor applied approaches or irrigation applied treatments. The treatment procedure, basically, there's an injection point where with CO2, you're injecting at 50 psi directly into the line after the non-return valve to sprinklers. Each treatment block irrigates at around 40 to 45 psi. Each line is equipped with a shutoff valve and a non-return valve, essentially to block any backflow system. And treatments are primed first into the line for about a minute. And after that, once it maximizes the pressure within the system, it's run through to do the treatment applications. In comparison, a tractor applied treatment would be running at around 15 to 25 psi pressure. We are looking at weekly applications of the chemical starting early March 2023. We are looking at a 10% incremental data, which is something we have learned over time working with commercial producers, that it could be a useful metrics to look at. We're also going to take Horsfall-Barat scale for 3 separate individuals who will be collecting that data weekly, and growers at the endpoint of the trial. We'll be doing assessments for each of the bed in a plot. We're planning to do mapping of the field with the multispectral sensors on drones to help us understand the canopy coverage and Alternaria leaf blight movement. And we'll be, of course, doing pathogen isolation during this period of time. One challenge we could face, this image taken by Derek, just to show that during high wind events, there could be some drift of the irrigation water in that system, which could limit its complete coverage within a small section of the outer 2 rows. In this particular, we are of course not doing this with our fungicide applications, but this is an observation we have seen. And to account for any changes within that structure, what we're thinking about is that each of the beds will be actually subdivided into 3 sections for rating. So in each treatment plot, we'll have about 12 data points for disease severity assessments, and then replicate it 4 times, just to add that statistical robustness to the study.
You have 1 more minute, Matthews.
Thank you. The schedule of the studies is that we plan to complete this Alternaria work by May, June 2023, and final reporting by June 2023. This helps us to also learn further in our setup on the cottony rot study. We did a survey in 2022. We did not find across any cottony rot on carrots, but I've already heard 1 or 2 instances earlier this week pertaining to cottony rot on carrots here. So we're trying to isolate and moving forward on a study for fall. With that, I'd like to acknowledge Ben, Mike, Dean, and Sudeep Sidhu, Dr. Sudeep Sidhu at Live Oak, a UFNF REC location, Darren Raj, Russ Hamlin at Grimway Farms, the California Fresh Carrot Advisory Board, and UC Cooperative Extension. Thank you. I'll take any questions if there's time. Thank you. What Bob wanted to note is that the fertilizer used in the study is a controlled release fertilizer from carrots, from Harrell's, targeted for long season release.
Okay.
Great.
Thank you, Dr. Maris. So our next presentation will be on screening carrot lines for resistance to cavity spot and other traits by Dr. Meredith McDonald from University of Guelph, Canada.
Great, thank you, Jazz. I'll share my screen. Wonderful. Yes, it's really nice to be here to give you an update on our work on cavity spot of carrots. Of course, you all know Phil Simon, and Umbrin Ilyas is the graduate student who's been working on parts of this project also. There we go. Our trials are done on very high organic matter soils in Ontario, Canada.
Good morning. Thank you.
Oh, sorry. Yeah, that might be a problem. Anyway, we'll continue. So we do have a good cavity spot nursery at this site. Each year the carrots are seeded in late May or more often early June, and were harvested in October. And you've already seen some good symptoms of cavity spot from Jaspreet. Our part of the work involves looking at the Pythium species that are involved in cavity spot. Work done in California over the years has found both Pythium sulcatum and Pythium violae causing cavity spot. Sulcatum is the main species we find in Canada, and it's also the main species in Washington State in their cavity spot nursery. And as we're doing some more molecular work, we're finding a little bit of some other species, intermedium, ultimum, and irregulari, but it's still at least 95% Pythium sulcatum. And this work is continuing both with isolations and with molecular identification. So of course, our objective all along has been to work with Phil Simon and to screen the USDA lines for resistance to cavity spot. We always have a highly susceptible line in our trials. Atomic red has been very susceptible. A lot of the purple carrots are highly resistant. Recently, we've been using deep purple. Then we have several cut and peel commercial cultivars, as you see here, including Imperial Cuts. Crispy Cut is a new one for this year. And then we include a number of cello carrots, including Maverick. Cello Bunch is the one that's very popular in our area. And Nairobi is one that's very popular in the UK and is considered to be highly susceptible to cavity spot. And of course, working with Phil, the long-term objective is to breed cavity spot resistant carrots and carrots with other beneficial characteristics for production in California. Our methods have been the same over the years. We assess around 60 lines. They're seeded on slightly raised beds in this high organic matter soil, 4 reps per line, and standard production except no fungicides applied for cavity spot. And we assess leaf blight. So you just heard a good introduction to Alternaria. Cercospora also occurs in our area, and we assess these together because they tend to occur together. And we've also been looking at forking of carrots, which can be caused by a number of Pythium species. We harvest in October, assess 50 carrots for cavity spot, assess another 50 carrots for forking, and the cavity spot is based on size. We look at disease severity, and we do that leaf blight assessment and also assess for bolting in the field. So in 2022, we had good stands in our trial, as you can see in this picture. Incidence and severity wasn't as high as last year, over 50, and severity of 23, which is relatively low. But we still had good separation of the carrots, and we could see the differences in cavity spot in the lines. We did irrigate in August. Irrigation, soil moisture helps to increase cavity spot, but then it was relatively dry in September and October after the irrigation lines had been taken out of the field. And there was moderate leaf blight, so we did get some good ratings on leaf blight as well, and a certain amount of forking. So our 22 results, you can see here, deep purple, the resistant, there was no cavity spot at all. Atomic red was our most susceptible. Interestingly, Imperial Cuts was quite susceptible in this line. But of course, the ones we're most interested in are these resistant, relatively resistant cultivars at this end. So a number of them were crosses with this MBH line, which Phil tells me is nematode resistant. And then a number of the other ones were all crosses with this line, F7738. So it's really nice to see this consistency with certain lines and low cavity spot. Just want to compare this to our 2021 data, where again, we had lots of the most resistant lines with this same resistant line cross. And again, this is the nematode resistant line, also looked highly or relatively resistant. So what do these carrots look like? You've probably seen these dark purple carrots, no cavity spot here. This is atomic red. So the ones down here have no cavity spot, highest cavity spot on this side. And then if we compare that to 2021, again, you can even see these large cavity spots, but about the same number of, same low number of carrots with no cavity spot. So these are the ones that are really interesting. The ones with the lowest cavity spot, you can see that they're pretty nice looking carrots. And if we look at the other ones, again, most of them with no disease and all pretty consistent color and shape. There we go. So we look at forking and we look at leaf blight as well. So again, deep purple had no cavity spot. These are the ones with lowest cavity spot. Forking was kind of all over the place, but relatively low in all these lines. And the one that's probably the most interesting, so the cavity spot resistant line crossed with the nematode resistant line. If you look over here, the low numbers mean low leaf blight. So also showing up as quite resistant to leaf blight. And again, if we compare that to 2021, we also had a line with the nematode resistance, relatively low cavity spot for that year. And again, relatively low leaf blight. So even though there was only moderate disease pressure, we had that nice identification and grouping of the relatively resistant lines versus some of the highly susceptible ones. We're focused on orange carrots, and a number of those had low cavity spot, and a number were crosses with nematode resistance. And then at least one of them looks like it has that ideal combination of low cavity spot, nematode resistance, and low leaf blight. We're still finding Pythium sulcatum as the main species involved in cavity spot. And of course, all of this is to contribute to Phil Simon's breeding program. Our reports are available online every year, and they're also provided to the carrot board. The 2022 results will be available on our website in March. And I certainly want to wrap up by acknowledging the funding from the California Fresh Carrot Advisory Board. And we do receive additional funding from these trials for some other sources. So I hope there's some time for questions.
All right, we just launched the poll. So again, if you need DPR credits, make sure you answer these two questions that should pop up on your screen. Mary, Mary Ruth, do you want to read these out for us?
Sure. What color of carrot tends to be the most resistant to cavity spot? And there's that whole range of carrots there. And question 2, what types of resistance can be bred into the same carrot line?
Awesome. So while folks are filling that out, we do have a question in the chat box. Is rating for cavity spot on purple carrot challenging?
Yeah, that's a good question, because we do get that from time to time, that maybe we just can't see the cavities. So there's two answers to that question. Phil has found a carrot line that's mostly purple and that's very susceptible to cavity spot. So it's probably not directly related to the purple carrots, but the short answer is that we can still see cavities on those dark purple carrots when they develop. Yes, just about everybody got the right answer, that the purple carrots tend to be the most resistant. And I can't see all of the answers to question 2.
You should be able to scroll down on it.
Oh, okay, right.
There we go.
There we go. Yes, and again, all of the above. So it's looking very promising that Phil can breed for resistance to cavity spot, alternaria, and nematodes all in the same line.
Great, thank you.
And I'll stop sharing.
Thank you, Dr. McDonald. We will move on to our next presentation, which is carrot breeding to develop and introduce improved cultivars for California production. Field research combining genes for root knot nematode and cavity spot resistance by Dr. Phil Simon.
Thanks, Jess. Can you hear me?
Yes.
Yes.
All right, good. I appreciate the opportunity to give you some results of our activities in the last year that have been supported by the California Fresh Carrot Advisory Board. I think this is my 3rd slide. I had turned over the slide. There we go. So not to correct Mary Ruth too much, but it's the USDA breeding program I'm talking about, but this is definitely a group project, and the input from all of these cooperators is not just helpful, but essential for the progress we're making in the breeding. So this is a joint breeding program, and all of these individuals listed here and more are involved. And I surely appreciate that cooperation. Next slide, please. The breeding program covers a range of things and things that are covered by some of the CFCIB funding. So I'll be rather brief with some of the different presentation aspects today. I'll be talking about our general breeding, which Jas is leading the collaboration on, our nematode resistance breeding, which Phil Roberts is collaborating with me on, some cavity spot and alternaria breeding with both Jas and Mary Ruth. And what we do with this project is evaluate materials in our fields, in USDA grown fields, and send those carrots back to Madison for lab evaluation and seed production. Then we get some roots from Dr. Roberts, from Phil Roberts' program for nematode resistance, and results from Mary Ruth and Jas's programs for the cavity spot and alternaria resistance. We're also doing some additional molecular marker work and quality evaluation for these carrots. Next slide, please. So first, to talk about the general field trials, our primary focus is carrots for California. So the University of California trial in Desert Research and Extension Center in El Centro, which we're going to be harvesting March 7th of this year, is the lead trial. We had 250 entries in that trial last year. We also have a trial here in Wisconsin and evaluated a smaller number of carrots, but many of the same ones that can be grown in California grow well in Wisconsin. And then we look at these materials, and we make experimental hybrids with them to see how the USDA hybrids are comparing to industry hybrids. And so that trial includes both USDA materials and industry materials. And we're quite successful at having some quite competitive hybrid combinations in both the baby and cut and peel carrot categories in our trial, but the seed industry certainly takes the lead on this program with providing growers with seed. Next slide, please. So our nematode work, shifting to nematode resistance breeding, is done with Phil Roberts Some evaluation is now being done by Mary Ruth and others on this at this meeting. The most recent large trial we had, Phil will talk more about, but it was harvested in August, had over 500 entries and was focusing on the Meloidogyne incognita nematode. And we look at quite a few sources of resistance with the idea of combining several of them to be able to have a more reliable and durable resistance. This last year, we didn't have a field day because of COVID restrictions. Next slide, please. Looking at the nematode resistance work, we have been making advances, continuing to make advances with the nematode resistance, which Phil and I originally found in the Brasilia cultivar. This is holding up for both of these root knot nematode species listed here. And in addition to this resistance source, a primary source that's newer than Brasilia is one from Homs. And Mary Ruth talked about this NBH 2306. That means it's a carrot that has genetic background from both Brasilia and Homs. And that's got some very good nematode resistance. And that carrot will be released this year to the seed industry and researchers to make that available for breeding resistance by that seed industry. But Brasilia derivative 3271 is also being released this year. We're putting in quite a bit of time evaluating some new sources of resistance, primarily one from South Africa, Cape Market. And we are including these nematode resistant carrots as part of our experimental hybrids in the breeding program. Next slide, please. So I will mention that we invite seed industry to provide seed to the nematode trials, and we have had participation from up to half a dozen seed companies, and some of them have some very good nematode resistant entries. We don't know their background because we don't ask them to divulge that, but we are pleased to see some good progress with the seed industry in making nematode resistant carrots. Next slide, please. This is a complicated slide that I'm not going to go into any detail about, but just to point out that we now are looking at, if you look across the top of this slide, 8 different genetic sources of nematode resistance. I mentioned a couple of them, and we're looking at pyramiding more of these genes to look at them both in the breeding lines, the parental stocks, as well as the hybrids, which are listed at the bottom of the slide. So this is just a demonstration of the fact that we look at a wide range of resistance sources to see which ones work best. Next slide, please. And so we start with carrots like the Brasilia, where you can see genetic variation for resistance in Brasilia in upper left. We develop inbred lines, release them to the seed industry. We make experimental hybrids with more of a cello shape in upper right and the baby shape on lower left. And then we have some quite good looking hybrids from these nematode resistant sources. Next slide, please. Moving on to cavity spot. Mary Ruth gave an excellent overview of where we're at this year, and the results are quite similar to last, which is always reassuring when you're in a breeding program to see the resistance is holding up. We will be releasing these 2 cavity spot resistance sources this upcoming year, 77, 37, and 38. And we'll also be releasing the nematode resistance source, which is a pretty good cavity resistance source. So these will be of interest to the seed industry for their use in developing new cultivars. And again, like the nematode resistance breeding, we're earlier in the process, cavity spot resistance, but we're looking at pyramiding or combining sources of resistance from different sources to make it more durable. Next slide, please. A couple of slides on alternaria leaf blight resistance this year, which we've had a little larger focus on. The USDA program looked at about 200 breeding populations, and those materials were derived from about 900 diverse carrots starting several years ago. So we're looking at a broad range of genetic variation to advance alternaria resistance. In addition to the trialing we do in Wisconsin, Mary Ruth mentioned resistance evaluation in Canada and JAS in California. And Mary Ruth combines this with her cavity spot resistance as well. I'll give you a little bit of data in the next slide where I'm presenting some information on 20 of the sources of alternary resistance evaluated in California and Wisconsin. The correlation between the scores in these two locations was a little lower than I expected, about 0.56. But just to go along with the other resistance breeding programs, we're intercrossing sources of the resistance as we go along. Next slide, please. I'll show you some of this is too much to talk about in any detail, but you can see here that the scores range from 0 to 50. I adjusted my scoring system to coincide with JAS's. So, excuse me, the scores are between 0 and 100 to coincide with JAS's scoring system. You'll notice that nothing has a score of 0 and not much less than 50 in Wisconsin, where the where the alternaria attack is very severe, and some carrots are pretty much killed totally topwise with alternarias. You can see with those entries with values of 100 in the Wisconsin score column. The California evaluations have a less intense alternaria attack on those carrots. Again, I mentioned in the previous slide, there's a fairly good correlation between the two locations. We find it takes several years of evaluation to get good alternaria rating score with the breeding material. So as we advance this, we'll be looking at both of our Wisconsin and California scores, but also next year I'll be sure to include more of the material we're growing in Canada to have Canada in the report we talk about next year for alternaria resistance. Next slide, please. So in addition to the resistance breeding and other things that we have ongoing with the project, we take materials from Phil Roberts' program, for instance, and also from our breeding program, and grow them in the greenhouse after they've been vernalized so that they'll start to flower. And that's what you see in the upper left-hand picture here. So we get seed off the carrots. We also do larger scale seed production as shown in the lower right. Because in order to keep the whole program going, we need to have seed, and it also helps us provide some materials for the collaborators that are so important for the project to be making advances. Next slide. I think we maybe have just sort of a final slide on terms of where we're at and what we're doing with things. We continue to look at new combinations of resistance sources for these three major diseases. We do have some effort on other diseases I didn't mention, but these are the big three that are primarily of importance for not only California, but really global carrot production. And as I mentioned before, we get both information and roots from the collaborators in the different trialing that's underway, and this is essential for us to make progress in the breeding program. In the long run, we're looking for releasing genetic material to the seed company, and we have a number of those releases coming up for several of the traits I mentioned. And as I also said, we'll be including some of the material so we can include Canada in the alternaria comparisons next year. Along the way, we are developing genetic maps to help us in the seed industry breed for these traits in the breeding programs and to develop more efficient breeding approaches. And the California Fresh Carrot Advisory Board funding is essential for our ongoing progress in the breeding program, and we certainly appreciate all their assistance. We have funding from other sources as well, but what I've talked about today is the focus for the CFCIB funding, and appreciate all of you for listening to my presentation.
All right, thanks, Phil. We have his poll up, so if you don't mind, Phil, just reading that question off for us.
Question here is genetic resistance could significantly reduce the need for applying nematicides. Is the primary question for today. Should be able to see all the results in one shot. So I guess to go along with the poll question, I will mention that as Jas had some trials with different nematicides, I think it would be good if we include some of the resistance material in that trial, Jas.
Yes, so we have plans for this year. Yes, we will be having those. Yeah.
Always some more things to do, but that's what makes it fun.
Okay. All righty.
All right. Thank you, Phil.
Thank you.
So we will continue with the same topic with our next presentation, identification of gene sources for resistance to root knot nematodes attacking carrots in California by Dr. Phil Roberts, UC Riverside.
Okay, good morning, everybody. So glad to be here. You've heard a little bit about nematodes and nematode resistance breeding. So we've had a good introduction from Phil Simon just now, and Mary Ruth mentioned some of the overlap between nematode resistance and some of the other diseases. I'm focused on identifying resistance sources to root knot nematodes and then bringing those into breeding materials to ultimately have good looking, commercially acceptable carrots for the carrot industry here and consumers. We collaborate very closely with Phil Simon, as you've already heard, and I'll highlight some of that. Bill Matthews in my program is instrumental in terms of a lot of the technical assistance and expertise in nematode screening. So I've shown this usually every time, but it's just a look at nematodes related to roots. In the lower left corner, you can see these stained second stage juveniles, and that is the stage that moves towards the root, penetrates into the root, and then moves within the root and establishes a feeding site. Over on the right-hand side, you can see swollen female, bottom right hand, this white pearl-shaped structure with a big blue egg mass full of several hundred egg masses attached to the female, and this whole process causes the disfiguration of the carrot root, both the fibrous root system and the taproot, forking and the galling symptoms. So we have worked for many years, initially identifying a source in Brasilia, the MJ1 resistance gene, source of resistance. It's dominantly inherited, and since then have looked at a range, these are indicated in blue, a range of materials from around the world: Homs, Pingding, Scarlet Fancy Favorite, and so on. A couple in red in the middle here, Cape Market, and also PIs or planned introductions represent newer sources of resistance where we have excellent strength of resistance, multiple genes conferring resistance, so opportunity to combine resistances into a durable platform that will be effective in the field for the long term. Just highlighted at the bottom, combinations of the Brasilia resistance with the Homs resistance are looking exceptionally good and will be in some of the release materials. And then we have at the bottom a Homs selection that is has very strong resistance to Maloidogyne hapla, which we find in cooler areas. Most of our focus has been on Maloidogyne incognita, which is the predominant nematode in the warm soils of California and also anywhere else carrots are grown. But we have other species such as Javanica, which are also found and important. Our trialing this year was held at the in the field was held at the UCR Coachella Valley Ag Research Station, located in Thermal, just east of Palm Springs, just at the northern tip of the Salton Sea. And we had a very strong Maloidogyne incognita infested block that we've been managing with growing susceptible crops in the years preceding the trial year, tomato, sorghum, and also a bean, lima beans typically. So this builds up the population. The planting here was made in late March 22nd. The harvest and assessment was done in mid-August. Was a very hot year. We had several heat wave events, and it's quite remarkable actually how well the carrots grow with a combination of nematode infestation and high heat. But the trial was actually very successful. Here are young carrots coming up in April. In the trial site, you can see just to the left of sender, we have nutgrass problems, but we've done pretty well controlling that with herbicide. We've had good advice from John Gerard and others in the industry, so we try to keep on top of that. I'll just mention at the Coachella site, there's another block we have established now with Meloidogyne javanica. So we have incognita and javanica sites at the Coachella site. Part of this was motivated by the fact that UCA&R has sold off the nematode fields at the South Coast Research and Extension Center, so they will no longer be available to us. But anyway, we've put some of the project funded effort into establishing a javanica field. Here's an example of incognita symptoms in the Coachella trialing site. You can see the extensive galling showing particularly on the fibrous roots. Here's a close-up of that. So we have very strong infestation levels based on our multi-year management of the field. As I said, assays done in the third week of August, very, very hot, done over 2 days. Some of the industry folks also came and looked at their materials. And what we've been putting in the trials is a sequence of selections that come out of earlier years. So out of, and I just, as an example here on the left is a Brazilier Homs combination of resistance, sorry, on the right, a resistant carrot entry grown in previous years at both Kearney and the South Coast field stations. And on the left, there's one of our susceptible Imparata 58 checks, which we grow every 5th plot throughout the row. Here's another close-up, an F4 with a Homs and Brasilia background, in which Phil has done an excellent job of getting the resistance into something that's looking like a good long carrot in terms of agronomic quality, which is really for us, is the stumbling block in terms of getting variety releases, is to get the resistance into something acceptable. So here's a quick summary of the field evaluations in the last year. These are tabulated in detail in table 1 of the report to the carrot board. So for those in the seed companies and breeding programs, please take a look at that. We had over 600 plots, including the checks. This is material primarily from Phil Simon's program. Also included several hundred industry plots that were either integrated into the main plot trial area and in some dedicated rows where we had industry participation within the infested site. The routes selected, as Phil had indicated, were shipped with desirable traits, looking resistant and good carrot quality, shipped back to Wisconsin for seed production, selfing and or crossing. We had up to, in advanced plots, and you'll see in table 1 the details, we had up to over 80% of advanced lines with really good resistance. These are on our goal rating score of 0 to 9. These are looking at scores of less than 2, many rated with 0 under very heavy infestation. On that scale, the Imperator Czech mean scores were 6.62, very similar to 2021, where we had 6.89 as a mean score. We had a lot of early generation progenies, one with some with Cape Market, where Phil and I are working with a graduate student who's looking at the incognita resistance in Cape Market. And so that's a summary of the field data. Please look at table 2. Just quickly then, also in the report, we describe several greenhouse-based experiments where we're looking in detail at the genetic control of resistance to help in the breeding program. Here's a typical greenhouse setup. The same setup looking later, carrots grown for about 2 months, inoculated at the 3rd to 5th true leaf stage, and worked on drip irrigation in a sandy soil. Works very well. And just to summarize there, a couple of points. As I said, this is summarized in the report to the carrot board, looking at Cape Market derived populations and also some of the new plant introductions where we have multiple root knot nematode species resistance. So Incognita javanica, Arenaria, and Hapla. Some materials have combined resistance to these species. Others are strong against 1 or 2, but not others. We're continuing to work with genetic mapping to Meloidogyne hapla in HOMs. A selection of HOMs have a major resistance locus identified on chromosome 9, a minor one on chromosome 2. So this provides markers for selection. We're doing more genetic mapping with incognita resistant populations. And as Phil indicated, we've been reselecting for 7 released resistant inbreds. These are the NB, NS, NH lines, and then testing best resistant parents for hybrid releases that Phil had mentioned that hopefully will be coming out this year. So with that, I'll finish and recognize the funding from the California Fresh Carrot Advisory Board and also USDA funding, our interaction with seed companies, which is vital. And just to indicate our planning for this year will be a Coachella again scheduled for late March. So with that, thank you very much. Yeah, so I'd asked you which root knot nematode, Meloidogyne is the genus, which species is most common in warm California soils? Well, the suspense is killing me.
I know. All right, I think we can go ahead and end it and share those results. Perfect. So
Hey,
yeah.
well done, everybody. It's complicated because for a particular grower, javanica might be the major problem. But if you look across geographical areas, incognita is more prevalent, and that's why we tend to, in recent years, have focused more closely on that. Thank you.
Thank you, Dr. Roberts. We will move on to our next presentation, which is the root knot nematode injury prevention in California fresh carrot production by Dr. Oli Becker and Antun Flo, University of California, Riverside.
Okay, so this is a collaboration between my lab and Antun Flo's lab. We are working with nematodes. We had a pretty good introduction. We are always the last group to talk about nematodes, and so some of the slides might be similar to what Phil is telling you. But I like to show these for those participants that are not so familiar with it. We have several symptoms on carrots caused by root knot nematodes. The stunting could be early in the season when the population is very high. We have galling and forking. And of course, one of those problems with forking is not only the physical damage, but it also brings a lot of soil into the production area, and so that's another issue. Root knot is of course not the only problem in carrots, but in California we really have only maybe Longidoros down in the Imperial Valley, and in some cases there are some minor problems with stunt nematodes. But overall, it's really root knot is by far the biggest problem in carrots. The life cycle, Phil already showed it. We have a penetration by the J2, by the second stage juvenile into the root tip. Root knot typically does not go in the older parts of the root. They go in the root tip, and then they create a feeding site. The root starts to swell, causes the gall, and the nematode goes through several molds, and the female swells up and finally breaks through the skin of the root. And we see here some stained, a stained female with a stained the offspring of the nematode, the eggs, and we can also stain the gel matrix in which these eggs are laid, and I'm showing this to really show that for control with nematicides, it is important either to hit the J2 before they enter the root, or later in the season there is a potential that a nematicide might influence the reproduction, killing the eggs. But once the nematode is in the root, they're basically home free. And we have, over the years, worked at 3 different stations in the north. That's the Kearney Ag Station. But unfortunately, in the last few years, we lost the support there. Many people retired, were not rehired. So when the pandemic came, we had to drop those trials because we had to separate our team. Each of us had to drive by our own car, and that was just financially no longer possible. And then with the lack of support from the station, it became not feasible to continue that work. The one in the middle, that's where Jazz is now working. We used to work in the past with Joe Nooning, and we'll try to pick up with Jazz again. It's some collaborative work. And then finally, down in Orange County, that's our main station in Tustin. Phil already mentioned our division, in their wisdom, decided to sell our research plots, make space for a few thousand students. I'm sure the people along Irvine Boulevard will be very happy about this. We kind of found another spot on the station where we are in the moment trying to re-establish our plots for the nematode work. Let me say something about the past 10, 11 years. We have tested approximately 20 synthetic compounds or products, about 11 different microbes, microbial compounds, or and 4 botanical products. Most of these products we've tested at least in 2 years to get some significant results. And there are many trials we ran that we usually don't report here because it would be just too boring, because most of these compounds did not have any significant effect on the nematodes. So the objectives for this year were that we continue to compare the efficacy among a new generation of nematicides. I like to say we are now in generation 3 for non-fumigant nematicides. Fumigants we haven't tested in the last 10 years. The writing is on the wall. We just heard from DPR again some increased registration requirements or use requirements for Tilone. And so we were really concentrating on non-fumigant nematicides. And we also have seen in the last few years there are application modifications we can use to improve the efficacy of the development products, and we will follow this up also in the next year. The new nematicide generation, I have the representatives here. So that is fluopyram, vellum from Bayer, a very effective nematicide in vitro. In vivo, it has some problems in motility. It does not go very deep into the soil. NemaStrike was a product from Monsanto. They stopped a couple of years ago distributing it. It was already registered as a seed treatment in various crops nationwide, but they have run into problems, and the whole issue with Monsanto and Bayer, it's an iffy thing anyway. Salibro, that is the old, well, I shouldn't say old, the product from DuPont. We have worked many years now with this, and it looks now that perhaps this spring or summer they will have registration, federal registration, and the California registration should actually come very soon. Then Nimitz done by Adama. It has a registration. Jazz mentioned it. It is problematic in terms of when to apply and how to apply it, especially if you don't wait let's say 1 to 2 weeks after application. If you seed the fields, there is a delayed emergence, and so look at the label. There are some requirements, and that's important. Timurium is the latest development from Syngenta. It is a very similar compound to vellum, highly active, has also some fungicidal activity, just like vellum, but it is still years down the road from being registered. Our trial setup, usually we have a sandy loam. We try to shoot for an initial infestation between 10 and 30 J2 per 100 cubic centimeters. That is not always easy to do. We manage our fields the whole year round to have a very even infestation. That's why most of our trials are also on stations where we can manipulate those fields all year long. We have complete block design, 5 reps. The products, we typically apply broadcast on top of the beds and then incorporate it with a rototiller. And I come back to this because that is really the key for an excellent control of all of these compounds. We look at the galling after 7 weeks or sometimes after 12, and then determine marketable yield, number for carrots, and also the weight. This is my scale, marketable, non-marketable, everything 0 to 3. I just subjectively determine as marketable, and the rest is non-marketable. We use, like Phil and Phil, the Imperator 58 as a check. I'm not going into details here. This was what we published a couple of years ago in multi-year experiments. We pretty much nailed it down to a check. This is the green green column here on the right side. We will have an infestation that gives us a rating of about 6, similar to what Phil just showed us. Then Nimitz, significant effects, but vellum, those are basically in the middle of our field, and then Salibro, certainly the best compound among this group over the years. And you see here on top we have in various years very high infestation. I should really point out, you know, these are experimental conditions where we want to have a very high infestation. The growers among you probably would not go in a field like this. This is just to have a very, very high disease pressure. And give you an impression how this looks like. On the left side, the non-treated control, and then we have Salibro, Nimitz, and vellum, and these are, yeah, I would say an average of what we see over all these years. Here we have another vellum. Oops, sorry, vellum treatment. And this was the first year where we tried to improve the application of vellum by adding soil wetting agencies, and here we have Absorb and Watermax 2, and we saw a dramatic really increase in the percentage of marketable carrots, and you see here on the bottom left is a control, really pretty ugly, and on the right side one of the vellum treatments, and what is very characteristic for vellum, if you don't have the wetting agents, the last 2-3 carrots here on this picture on the right side shows you about the depth of a normal vellum application, the 3 to 4 inches deep, or sometimes even less, depending on the depth of the incorporation with the rototiller.
You have one more minute.
Thank you. There is a Salibro. We had in this year, we had a very low infestation in the beginning, and consequently we had a tremendous result with Salibro and compared to the control. a high marketable increase. Finally, in this year, I should skip this one and go one next to it. This year we we had the WaterMax 2, this soil wetting agent, in with all of the products, and in all of the products it increased the percentage of marketable yield. So, and that's what we'll follow next year or this year if we get our station established. Thank you very much. Okay, what is the primary reason to combine non-fumigant nematicides with a wetting agent? To reduce the dirt covering the carrots at harvest, or to make it easier for the carrots to grow in heavy soil, or to promote soil penetration by nematicides, or to repel gophers from eating the carrots?
All right. I think we can go ahead and end it and share the results. And if you don't mind, looks like majority of them have the same answer.
Exactly. Excellent.
Very good. All right. Thanks, Oli.
You're welcome.
Thank you, Oli. Alrighty, it's 10:35. Welcome back, everybody. And we will continue with our presentation. And our next presentation is Steam Disinfestation of Weed Seed Banks in Carrots by Dr. Steve Fenimore from University of California, Davis. Steve?
Well, good morning, everybody. A little shifting gears. So I'm going to tell you about some work we're doing. It's a sort of the interface between engineering and pest control. So we had a couple of objectives. We're working on steam and carrot for control of soil-borne diseases and weeds. We have a lot of activity on weed seed, as well as Pythium, sclerotinia, which is a major pest in lettuce, and a number of other diseases. So we tested a new machine this year, and I'll show you some pictures of that. And we did not, at the beginning of the season, did not know the ideal bandwidth or depth of treatment for steam application in carrots. And we did this in experimental situation. We really need to do it in a commercial situation. So I'm going to use the word a few times, soil disinfestation. So what do I mean? It's just a general term that we use to talk about reduction of the pest community in the soil to a level that will allow the crop to thrive. It's a kill step. Fumigants are an example of a kill step that reduces the pest population to tolerable levels. You can also use heat, solarization, or steam. And we'll talk about steam. I talk about a engineering term called the dwell time, and that simply means the amount of temperature over a critical range, and that's 158 Fahrenheit or 70 degrees centigrade, if you like. So that's our cooking time. Now, the machines that we use are continuously move. What we find is that if we heat soil up to a critical point, it tends to cool very slowly. So the machine is moving continuously, the soil cools slowly, and it allows sufficient time to kill the pests. So it's a machine, and you can do lots of different things. You can do an endless number of possibilities of applying steam, shallow, deep, broadcast, or band. That's what we're doing here. Broadcast, where you're treating the entire field, and then band, where you're just strategically placing it where you need it and not treating where you don't. So how does it work? You inject steam into the soil to raise the temperature to 158 for 20 minutes. Again, the dwell time. And so what steam does is basically just using water molecules as a means of transferring heat from a burner, a heat source, to the target soil particles. And when steam comes in contact with these much cooler soil particles, the steam molecules, they condense and they release their heat, the heat that they've carried from the burner down into the soil. And any pests, be them weed seeds, nematodes, or pathogen propagules, are killed by the heat, providing they're exposed for a sufficient amount of time. Incidentally, I've done a little work in a greenhouse, and root knot nematodes are pretty darned easy to control with steam. Steam also kills the pathogens in and around the soil particles, and it can also cook nutsedge tubers. So why choose steam? So it's a complicated mechanism, machine-wise, to deal with it, but this is one reason. These are some of the reasons why I continue to work on it, because I see potential for commercial application of it. It works. It kills soil pests. Nobody owns it. You can't. It's not like a pesticides, which is somebody else's property. It's a device as defined by US EPA. Hose are devices, fly swatters are devices, insect traps and mouse traps and such. Those are all pest control devices. It's a sanitation treatment as defined by the organic standards. So it's organic compliant. It's flexible and it's safe. As long as you don't touch the hot surfaces, it won't burn you. There are no buffer zones, no township caps, no notifications, and there's no plastic mulch needed. Our treatment time or covering it is just a few minutes. So what are we doing? A little bit on the logistics here. We're injecting steam into a targeted area. You can make all kinds of combinations of adjustments. It's metal, it can be changed. And so what the idea is that you go in with a pre-plant treatment, you treat the band, and then you plant the crop, and the crop grows. So what we did in 2022, we tested this machine. We did basically the trial number 1 is basically a shakedown cruise to make sure that this machine works and we learn how to use it. And so what we did, we only went 2 inches deep by 4 inches wide. So the width was only 2 inches. I'm sorry, the width was 4 inches and the depth was only 2. And these are on 80-inch beds, and we're treating 2 of them at the same time. Then in the second trial, which we started in early September, we did it with the same unit above. We did not at the beginning know how wide we need to be or how deep we need to be. And so we had 2 bandwidths, 3.5 and 5.5 inches wide, and 2 4 and 6 inches deep. And these are all replicated trials. So here's what it does to the soil. So this is just showing the 4-inch wide bands. And we ended up really liking the 4x4. It seemed to be the best combination, because by injecting the steam at least 4 inches deep, it tended to maintain the heat for a longer period of time. So again, what I'm going to put on my laser pointer here. So what we want is 20 minutes of dwell time. So we want that above 60 degrees centigrade. So that's 140 some degrees. I don't remember exactly. We maintain that for over 20 minutes by injecting 4 and 6 inches deep. Really, the 4 did quite well. So this is what the weed control looks like. I'll let you look at that for a little bit, because basically there are very small carrots in here. You can very hard to see them, but this is the idea. This is on the research station, and we have a lot of weeds, as you can see. And this is what it does. And we're getting very, very high levels of weed control. For the most part, we're getting close to 100%. So these other weeds are easier to control. And here, this is the same setup in lettuce. Lettuce plants were a little bit larger, a little bit more mature. So purslane, we got 99% control because purslane is a shallow germinating. So we're killing the seeds, and the seeds come up from very shallow layers in the soil. We're getting shepherd's purse, nettleaf goosefoot. Goosefoot is kind of an interesting case. Burning nettle, henbit, pigweed, 100%. Little mallow is a little tougher. It comes up from deeper in the soil. So it's a bit more difficult to control, which is not surprising because it's not easy to control with fumigants either. So we collected the data, weed densities, hand weeding time, because we're interested in the economics. We need to pay for the treatment by reducing labor input. That's sort of the logic here. We measure Pythium control. Pythium, because it's an issue both in carrot and lettuce. We're working in both crops. We're measuring crop yield. And then the treatment list is very simple. It's steam versus no steam. And again, these are replicated trials. And so here's the weed control by species. So I'll point out the highlights here: purslane, shepherd's purse, burning nettle, henbit, sow thistle, and groundsel, all very good control. Puzzling, goosefoot, not such good control, and hairy nightshade. So there's a bit of an interesting backstory there. Malva, I won't say much more about that. So let's see, the hand weeding time. No, wait a minute. This is with the weed control I can't see the whole thing because this share thing is in my way. So this is hairy nightshade. That's right. So hairy nightshade, we have to get the steam deep enough. We have to get it at least 4 or 6 inches deep. This is 2-inch wide band. This is a 4-inch wide band, which gave perfect control. So we need to get it deep enough. What that's saying is that those weed seeds are coming up from as deep as 4 inches, which is a little bit deeper than we expected. Reminder, that's hairy nightshade. So this is Pythium control. So this is reduction. So we want a tall bar. So first I'll point out the control, which was not suppressing Pythium. And we use Pythium both because it's important, moderately important, but also because it's very easy to control with steam. And so if we're not doing a good job in controlling Pythium, then we know we're doing something wrong. And so when we get down to 4 to 6 inches deep, basically, regardless of the bandwidth, we're getting good Pythium control. Hand weeding times. So what we want here is the bar to be as low as possible. So these are 3.5 inch wide bands, 5.5 inch wide bands, and they're at different depths: 2, 4, and 6. Again, 2, 4, and 6 inches deep. The control, I didn't put the control bar on here because it kind of distracts, but it would have been shooting through the top of my computer. It was because we had so many weeds, 386 hours. So basically what this says is that we actually got the best weed control at 6 inches deep with a 3.5 inch band. And I do not understand this and can't really explain why this particular band was worse. So it's basically the opposite. As the deeper we go with the wider bands, the harder it is the longer it takes to control the weeds. Probably we're diluting our heat too much. That's my best explanation. So we did some preliminary cost studies back in, just a reminder, and I need to repeat these again, and we're working on that. But this is probably a bit of sticker shock. We think we can cut this in half. I'm pretty confident that we can, and so something like $500 an acre or less. Again, we're trying to use this to reduce labor inputs. So I did not submit a proposal to the fresh carrot board because we got some additional funding from the USDA NIFA and from the PERC, which is the Propane Education and Research Council. And so I'll tell you what we're doing right now.
You have about a minute left.
We are building a commercial scale steam applicator, and that's what we're working on now. And with our existing applicator, I do want to come to Kern County and at least get a couple of tests out. So what we're doing on the new applicator, we're working with a French company which makes a very good boiler. It has an onboard preheater and a superheater, which allows it to disperse better in the soil. So I'm working with Peter DeGroot and Mark Siemens, 2 engineers, and what we're doing is preparing to build. We're heavily into it, ordering the supplies and such. That'll be on tracks, and we'll have a bed shaper behind it. And so Peter's working on getting the specifications so that we can get a fine diagram of that one. So that's what we're up to. Okay.
All right, great. We'll go ahead and launch your poll here. I think you had 2 short questions.
Okay, so true or false? Steam treatment of the soil is a patent protected process. True or false?
You can read the second one too.
So the second one, treating soil for 20 minutes at 158 degrees Fahrenheit is what? Is it pasteurization, sterilization, or is it neither? Or is it both?
And while folks are answering that, we do have a question in the chat. What is the volume of the steam unit? How long can it run?
The volume of it?
Yeah.
The one we have now? Well, I just got a 500-gallon tank to tow behind it, so we can go the length of my goal is to have enough onboard water to run a quarter of a mile. So yeah, we will be able to run. Well, I think some of those carrot runs you guys have in Kern County are even longer than that, but that's what we're doing. So however, I'll have about 600 gallons of water, and that'll allow us to run for a couple of hours.
All right, so we'll end the poll and share those results.
Yeah. So true or false, it's patented. It's not, there's no intellectual property here there is some very old stuff, but what we're doing is not covered by any patents. So our ideal is to build a machine and put the specifications out there. So if anybody wants to build one or modify it, they can take it to their shop and build it. Now, the second one, I unfortunately didn't say anything about the pasteurization and sterilization. This is a Goldilocks treatment. We're trying to not get too hot so that we don't want to kill the beneficial organisms like the nitrifiers. We want to it's Goldilocks, not too hot, not too cold, but we need to be hot enough that we kill the pests. So if you oversteam, you will sterilize. You do not want to do that. You're wasting money and you're hurting the soil. Okay.
Great, thank you.
Thank you, Steve. Alrighty, our next presentation is inoculum preparation for research farm to measure the effectiveness of new biocontrol measures by Dr. Isolde Francis from California State University, Bakersfield. Isolde?
Good morning. I'm very glad to be here. I would like to report today on a collaborative project that was funded by the California Fresh Carrot Advisory Board to Dr. Sidhu and myself to start a carrot cavity spot nursery at the Shafter Research Farm in California. So we heard already a lot about cavity spot. Just very briefly, the pathogen creates small sunken brown lesions on the carrot tuber, and even a couple of those lesions can get the carrots rejected for the fresh and the processing market. And the disease actually occurs more often towards the end of the growing season, so that often leads to it being overlooked until harvest. Now, there are several pathogens that can cause the disease, and as Dr. McDonald said, in certain areas, you have different pathogens that are the dominant ones. So in California, we mainly find these three here. Pythium violae definitely be the most dominant one in California, in contrast to Pythium sulcatum that was reported by Dr. McDonald. These are oomycetes. Common name for oomycetes are water mold, so they like moist conditions. We actually have one real way to manage them with chemicals, and metalaxyl and mefenoxam are typically used, although we see that the results are often inconsistent, and people have attributed that to increased microbial degradation of those products in the soil. And also there's reports of pathogens that are becoming resistant to these chemicals. On top of that, these cannot be used in organic farming. So the disease actually remains very important to the carrot growing community all over the world. So it's a difficult to manage disease. It remains important. Some previous reports on problems with germination. So the team did not want to take a chance, and they double seeded the carrots. Even though they were very young, we found really nice carrot lesions on several of those carrots from the 6 locations. They were brought into the lab. The tubers were rinsed, and small pieces from the edge of the lesions were taken and put on selective medium. Now, might be a little bit difficult to look at because the growth is very faint on the agar and you have to hold it into the light. So I try to put it in a different angle to take the picture, but what you can see here, an early age and a later stage here, you can see the pathogen nicely growing out of the sample carrot lesion. So that means that the pathogen is active in the field. So that was a very good outcome actually of a first try. Jaspreet is already running trials in that field, but to keep active and to keep the disease pressure up, we want to inoculate every year again. On top of that, I actually want to mention that Pythium violae and also sulcatum have been reported to be slightly different in their life cycle than other Pythium species. In other Pythium species, the zoospores, which are spores that have flagella, and flagella actually are these things that stick out of the spore and are used to swim. And so they use the little water film to swim towards their host. That for Pythium, and those are, sorry to miss saying that these are the most infectious stages of most Pythium species, yeah, because they can actively swim towards their host. They can also sniff out their host, so they can go very directionally, and then they will infect the host. Now for Pythium violae and Pythium sulcatum, it's actually reported that most likely this stage in their life cycle is missing. And so that means that for those main cavity spot pathogens, infection will rely on mycelium infection, which mycelia, which are these long hyphae, the white mycelium that you saw on the rice seeds, it's kind of fragile. How does the pathogen survive in the soil? It's through oospores, and you can see that here. These are actually spores formed through sexual reproduction, and they can also use as survival structures. So oospores are way more robust than mycelium. And so we're going to try to now, instead of putting mycelium onto the field, put oospores onto the field, and when they germinate, as you can see here, they can then immediately infect the carrots. So we're currently doing that because oospore production takes a really long time. We found this really nice thesis from Catherine Hills in 2018 that she reported on several different methods on how to get these oospores, liquid medium and in a solid medium, which is a moist sand mixture with milled oats. And so we're going to monitor these and look under the microscope to see when we have oospores and try to optimize this method to then use this to infect the field every year to keep the disease pressure. Why do we want to get the oospores going? Not just for the nursery, but I also want to do that for my own research on cavity spot in the lab. So this is how we grow carrots in our lab in these tree seedling pots. It has been very difficult to mimic, what is going on in the field to get reliable cavity spot lesions. And so we hope that with the oospore method, we will get a more reliable method of getting the cavity spot lesions, because what we're working on is actually using beneficial bacteria like streptomyces, which are bacteria that actually look and behave like fungi, to use as a biocontrol way for cavity spot. So these bacteria have been known to be very common soil inhabitants, and they are known to produce a lot of antimicrobial compounds. And what we have found in our lab, and I reported this on this last year, we have 4 really great streptomyces species that can inhibit the growth of Pythium violae in the lab. We have also seen that, sorry, this is too fast. We've also seen that it's a secreted compound, so something that is soluble in water that is secreted from these streptomyces species. We saw that because when we cut out the streptomyces before we put the Pythium down, still have inhibition. And this is we tested if it would be a volatile compound that does the inhibition, which was a negative result. So it's a soluble compound. And so we're kind of a bit stuck here to test it in the soil, because we don't have a reliable method to create disease. So how can we then actually see if the method works? So in the meantime, we did this, what some people call a biocontrol disc assay, although I don't agree, but this is a carrot, of course, mature carrot tubers. You put discs on there. You can see here the outline of the pathogen alone, or the pathogen on top of just an agar piece, streptomyces alone, or the pathogen on top of the streptomyces. What you see here is that the pathogen on top of the streptomyces, there's no lesions there, where the pathogen alone or on a regular piece of agar gave lesions. So this has a small indication to say that they can inhibit lesion formation by the pathogen on the carrot tuber. But of course, we're waiting for our soil assay to be optimized to actually test it in the soil. And then, of course, we are hoping when we get good results in the lab, we can take that also to the nursery in Shafter and test it over there together with all the other products that Jaspreet and her team will be testing. With this, I would acknowledge, of course, Dr. Sidhu and Joe Nunez, who have been great mentors for me and great collaborators. This project on streptomyces is actually the main thing that I work with my students in the lab. started with the master student, Ms. Bashadri, who has graduated already, and these 3 young fellows here that are continuing her work. And funding, very important support from the California Fresh Carrot Advisory Board, and we also received funding from the USDA. Thank you so much.
Great. Excuse me. Great. Thank you. We'll go ahead and launch your poll now.
Right.
If you could read that off for us.
Yes. Which part of the life cycle of typical OMI seeds it's not observed for Pythium violae?
All right. And while folks are answering that, we do have a question in the Q&A. So have you tried vermiculite with V8 to prepare the
Pythium inoculum.
there it is. I'm not even gonna try.
That's okay. Yes, I know this is a very common method to grow Pythium. I have tried it in my lab and was not successful for it. I know a lot of other people, like Lindsay DeToy, uses it very successfully. I might have not used the right conditions, but for me, it was not successful. So that's why I switched to the rice seeds, which was very easy for me to do. And so I will continue with that. But yes, it is it's a method that works for other people.
Okay, great. So we'll go ahead and share your poll results here. And if you don't mind just talking about the correct answer, it looks like there's
seems
some...
that the answers was a little bit different, but it's actually the zoospores that are missing. So zoospores refers to spores that can swim in the soil and as a major inoculum form of most Pythium. And we are now shifting towards getting oospores as a reliable inoculum because they do make oospores.
Hi, Slowly, we have a question in the chat as well for you.
Okay.
It's like, can you please share the size of pot for lab bioassay? And do you assess cavity spot on carrots in lab bioassay? If so, then after how many weeks of seeding carrots in the pot?
Yeah, so the size of the pod, I don't know exactly by heart, but I can share that with you. Those are 3 seedling pods. I think they're about like 30 centimeters long, at least 25 to 30 centimeters long. We grow 1 carrot per pot. Yeah, it's a long process. Carrot growing takes a long time. So we grow them for 16 weeks in the lab because it takes 16 weeks to get a good sized carrot. And yes, then we pull them gently out and then we can assess for cavity spot lesions. But like I said, we had inconsistent results for now. We had very good start. It looked very promising. And then the repeats were a little bit disappointing. So we're trying to optimize it. But if you want to talk more about that, just send me an email.
Alrighty, so we will be switching gears here with our next presentation from Dr. Ali Montazar, University of California Cooperative Extension, Imperial County. And he will be presenting on the best irrigation and nitrogen management practices in the low desert carrot production system. Ali?
Thank you. So good morning, everybody. We started this study in 2019. The main objective was to develop more information and tools about best irrigation and nitrogen management in carrots in California. So far, we have conducted 4 trials at UC Desert Research and Extension Center. We work with 13 commercial fields in the desert and 6 commercial fields in Kern County. They had different soil types. I believe it can represent the soil types, the common soil types we have for carrot production system in California. We studied fresh market and also processing carrots and sprinkler and furrow irrigation in the desert and sprinkler irrigation in Kern County. So I think it can represent the production system for carrots. For the Desert Research Center, we evaluated 2 irrigation regimes and 4 nitrogen regimes. This is the plot plan for 2022. But for growers field, we selected experimental assigned area, which had homogeneous soil in part of the field that the soil dominate the soil types in the field. We selected 5 subplots in each of those areas, and all the soil and plant sampling and other measurements concentrated on that area. So here I developed a video, 1-minute video, to demonstrate what we did in this study, and also to look at the research plot and the field we worked with. I hope it works well here. Let's see. Okay, so let's start with the crop water use or actual evapotranspiration. So these two plots compare what we learned about Imperial County and summer planting in Kern County. You obviously see the crop water use, or what crop needs, the water crop needs, is exactly inverse in Kern County and Imperial County. So which means in Imperial County, the crop water needs start with a higher value, then during the late fall goes down, and again continuously by the harvest increase. In Kern County in summer planting, it's against, started with a low value, goes up, and then continuously reduced over the season by harvest time. So what we learned in Imperial County across these experimental fields, so crop needs about 11 to 19 inches of water. In Kern County, in the summer planting, crop needs about 10 to 13 inches. And in winter planting, it needs about 20 to 23 inches. So I categorize, what we have in California based on the water needs for carrots into 3 categories: low water use, medium water use, and high water use. Based on this, it shows, so the summer planting in Kern has the lowest water use we have in California. And planting in Imperial is medium water use, and winter planting in Kern is the high water use carrots we have. So I think it can help growers in the future. If we have drought condition, if we are under stress, we can follow these perhaps to reduce the winter planting in Kern and increase the low summer planting and fall planting in Imperial. So they should decide in those drought conditions, go for what planting method. So this is applied water in carrot fields in Imperial. It shows a wide range of applied water. We obviously see what's going on. But one solid conclusion here, I think our growers over irrigate during germination. Definitely, crop needs about 3.5 inches of water. In some fields, we applied about 15 inches or 13, 14 inches. So we over irrigate sometimes. But also we learn we can consume some water if we convert furrow irrigation to a sprinkler. An average of 0.7 acre feet per acre. It's I think significant in the desert region. So in terms of nitrogen, we didn't see any nitrogen deficiency in any of these experimental sites, but this is the trend. So in some field, they started planting with a high content of nitrogen in the top 1 foot. But the average is a little bit lower in the early season, and then goes up in the middle, and then down in the late season. And this is what we are looking. We need to leave the field with the lowest amount of nitrogen accumulated on the top soil. In the Kern County, the same. In summer planting, but a little bit different. In summer planting, we see a high concentration of nitrate on the topsoil in 2 of these fields, and then goes down by the harvest time. But what I found field was odd for me, because the entire season we observed a high level of nitrogen content on the topsoil in this field 4, which should not be like this. We need to reduce the nitrogen content on the topsoil in the late season. So what we learned in the past, sandy soils are really at high risk of nitrate leaching. And across this all field we work, I found in one field leaching, high leaching. We need to be very careful in these sandy soils to how we manage nitrogen. So look at what happened in this specific field. So the blue line shows pre-plant. So of course we have a high nitrogen concentrate on the top, and within the soil profile by 5 feet, we reduce the nitrogen content. When we leave the field post-harvest, we see a low level of nitrogen content on the top, but a high nitrogen content in the depth of 5 feet, which obviously shows a high level of leaching nitrogen. So this is the nitrogen uptake curve we developed for the desert. As you can see, a wide range of nitrogen accumulated in the root and the tops at harvest, but 2 conclusions here. We have a linear model for nitrogen uptake in the root, and it started from 60 days after planting and continues to increase by the end of season by harvest. But for the top, it gradually increased from 40 days after planting, goes up, and when we pass 130 days after planting, so it starts to decline and level off during the late season. This is what is happening. So it means if you harvest the field around 130 days, so crop never reach that decline, and it goes up continuously but gradually. This is the other result we got if we consider 160 days as a typical crop season for carrots in the desert. So 25% of the nitrogen uptake is happening the first 80 days after harvest, after planting. And 50% is happening the following 50 days. I believe this is the most critical period for nitrogen application in carrot in the desert region, just these 50 days, because 50% of the nitrogen uptake is happening here. And then 25% of the rest of nitrogen uptake is happening during the last month of a crop production season. This is the result of 2021-2022 trial in the desert. We had 4 nitrogen treatment from 140 pound per acre to 775 pound per acre. This is the wide range, about 2 times. So we also had 2 irrigation regime from 100% of the crop water needs to 125% of crop water needs. Basically, it means 25% over irrigating. This is the result. What we learned from this result shows there is no significant yield increase or reduction when we go with 100% ET or a good irrigation management and an increased nitrogen application, from 140 to 235. Not significant difference in yield. But when we go higher nitrogen application with the 100% ET, we have some yield reduction. It's very interesting. If you apply more nitrogen, you might see even less yield. But again, when we over irrigate, if we go with nitrogen application of 140, we also have yield reduction, significant yield reduction if we over irrigate, but we apply less nitrogen. I think this is the main point. And then if we apply more nitrogen, there is no significant yield increase or reduction. I think this is the main point. So this data shows the nitrogen application rate greater than 140 pound per acre couldn't have a significant impact on yield. In a well-managed irrigation field. I think that's the main point. If we do a good job with irrigation management, 140 pound per acre should be work for us. But sometimes the problem is we are over irrigating. If we do over irrigate, 140 pound is not enough. We need to apply more nitrogen to make sure we maximize the yield reduction. Specifically, it's happening in sandy soils. This is another analysis we did based on the two first year data. We found positive impact on yield, on nitrogen application on yield, but there is no significant relationship. I think that's the main point. Some improvement, but not significant. So let's see what happened here. We increased nitrogen application from 170 to 270. We got half a ton per acre more yield. Then we increased from 13 pound per acre to 270. We even reduced 2 ton per acre yield in this increase. So I thought potassium also is interest of the industry. That's why I also did some analysis for potassium, specifically for the desert, because the Colorado River has a low value of potassium content. So what this data shows, the nitrogen removal at harvest varied between 2.3 to 3.9 pound per ton of fresh carrot. At the same time, the potassium removal at harvest varied between 4.8 to 8.1 pound per ton. It seems so the potassium removal is 2 times of nitrogen removal. It means if we have at harvest 35 ton per acre, and we have an average of N removal at harvest equal 3 pound per ton. So the nitrogen removal is 105 pound per acre. At the same time, the potassium is 207 pound per acre, 2 times of nitrogen removal. So we developed crop managed carrot module for carrot. So this is the first time all growers can use this software. This is a free online decision tool for all growers. So you can optimize your nitrogen and water application for carrots now. So, but currently we have a trial in Desert Reg. So in this trial, we even evaluate one treatment based on what crop manage says. It means next year I will come to you and show you, if we follow crop manage compared to what we do or growers do, the result, what the result says, and that time we can realize how effectively crop manage can help our growers for carrot management in the desert. So with that, I would like to offer my special thanks to California Fresh Carrot Advisory Board and also CDFA FRED program. We have a fund from CDFA. So with the great growers who supported this project and allow us to do this research in their field. And thank you very much for your attention. I would be happy to answer any question.
So our next speaker is not here to present live, but he has a recorded presentation for us. And that's from Dr. Jeremiah Drung, Oregon State University. And his presentation is on, he will be providing an update on SCRA-funded bacterial blight research.
Good morning. My name is Jeremiah Dung, and I'm an associate professor in the Department of Botany and Plant Pathology with Oregon State University. I'd like to begin by saying that I wish I could be there to deliver this presentation in real time, but I'd like to thank the California Fresh Carrot Advisory Board for giving me the opportunity to present a recorded version of this slide deck. Today I'll be delivering an update on SCRI-funded bacterial blight research. Before I begin, I want to introduce our project team and our objectives. The first objective is to evaluate and identify sustainable IPM approaches for bacterial blight in carrot and carrot seed crops. This includes myself, Dr. Taifo Mahmood, and Dr. Jeff Chang from Oregon State University. Dr. Lindsay Dutoy from Washington State University, and Dr. Jaspreet Sidhu from the University of California Ag and Natural Resources. Our second objective involves modeling the aerobiology and epidemiology of Xanthomonas hortorum pathovar caritae, or XHC, in carrot seed crops. This is led by Dr. Rob Stoll and Dr. Eric Partijak at the University of Utah, along with Dr. Walt Mahaffey with the USDA ARS. Our third objective is to determine the nature and extent of Xanthomonas hortorum pathovar caritae colonization in carrots. This is led by Dr. Jonathan Jacobs at The Ohio State University. Our fourth objective is to assess the population genomic diversity of XHC in the United States, and this is led by Dr. Jeff Chang, along with Dr. Elizabeth Savory with the Oregon Department of Agriculture. The fifth objective is to calculate the economic impact of bacterial blight and the associated management practices on the carrot seed industry, and this is led by Dr. Gina Greenway of Greenway Consulting. So the first project that I'd like to talk about involves whether or not fungicides commonly used in carrot and carrot seed production have the potential to increase bacterial blight. This work had its origins in some previous studies conducted by Dr. Lindsay Dutoy, who observed that when she applied Tanos to carrot plants, bacterial blight caused by Xanthomonas hortorum pathovar caritae seemed to increase in severity. So in this trial, we wanted to investigate whether or not fungicides that are commonly used to manage diseases like powdery mildew and alternaria in carrot can potentially impact bacterial blight disease in carrot and carrot seed crops. To help answer this question, we conducted two greenhouse experiments, one at Washington State University and one at Oregon State University. In this trial, we applied either one of three different fungicides, Raleigh, Roveral, or Quilt, with or without maicoside. And then we waited approximately 2 weeks, and then we applied Xanthomonas hortorum pathovar caritae. And then we let the plants grow and observed disease symptoms and quantified the pathogen on leaf tissue. As you can see, plants that were inoculated but did not have any maicoside exhibited the highest disease ratings in both of the trials. Plants that were inoculated and had maicoside with or without fungicides tended to have lower disease ratings in both trials, and we did not seem to see an interaction between fungicides and maicoside, indicating that no, fungicides do not exacerbate bacterial blight, at least in this context. We're also very interested in whether or not we can use debris management to reduce Xanthomonas hortorum pathovar caritae inoculum in carrot seed production systems. We know that carrot seed crops generate a large amount of debris that does not get harvested along with the seed, and this debris can harbor significant amounts of the pathogen. In this trial, we buried 10 grams of debris in pouches and buried those pouches in field soil in 1-gallon pots. We then subjected those pots to one of three different temperature conditions, 39 degrees, 59 degrees, and 82 degrees Fahrenheit. And within those three different temperatures, we had either wet or dry soil moisture conditions. We also subjected some pots to field conditions with ambient temperature and soil moisture. As you can see, at the beginning of the trial, we started off with just a little bit less than 1 × 10⁵ XHC per gram of leaf tissue or debris. And after 1 month, we were not able to detect the pathogen when the debris was buried at 82 degrees Fahrenheit, either in wet or dry soil conditions. At 59 degrees, we were not able to detect the pathogen when the soil was kept wet, but we were able to still detect the pathogen when the soil was incubated at 59 degrees under dry conditions after 2 and 3 months. At 39 degrees, we saw a slight increase in the pathogen at the 1-month quantification point, but then the pathogen started to decrease at the 2-month point, and by 3 months we were only able to detect the pathogen in debris that was stored under wet conditions at 39 degrees. As you can see, this indicates that there are interactions between temperature and soil moisture in terms of Xanthomonas survival in carrot seed crop debris. But this does indicate that management of this debris, namely burial, can help decrease the pathogen in carrot seed cropping systems. Another aspect of Objective 1 is to identify novel bio-based compounds for Xanthomonas management in carrots. In our lab, we have observed many different bacteria from both carrot leaves and carrot seed that exhibit antagonism or inhibition against XHC in pure culture. So Dr. Taifu Mahmood has worked in his lab to isolate 3 pure active compounds from these bacterial isolates and characterize their chemical structures. In an agar diffusion assay, all 3 of these compounds were highly active against XHC, even more so than some antibiotic standards that he currently uses for controls. The pictures on the bottom left are showing that agar diffusion assay, where those zones of clearing indicate antagonism or inhibition of XHC growth due to the presence of one of these active compounds. Some of the next steps that Dr. Taifu Mahmood is going to be taking is to determine the minimum inhibition concentration of the active compounds against XHC, work to increase the production of these active compounds so that we can test them in the greenhouse settings on carrot plants, and also explore the possibility of improving the production of these active compounds through genetic engineering. And of course, we have more bacteria that we are sending to Dr. Mahmood to investigate and try to determine if we can find more anti-XHC compounds. Objective 2 is focused on the aerobiology and epidemiology of XHC. The picture on the bottom right gives a clear example of why we're interested in this. As you can see, when carrot seed is being harvested, large amounts of carrot seed crop debris are generated, which can become airborne and contaminate newly planted fields or even the seed rows next to the beds that are being harvested. So for this experiment, we built a novel passive sampling device called the cascade settling trap, which has an opening at the top of the device and allows airborne particles to fall through, and they get sieved through different steel mesh filters at 400 microns, 150 microns, and 50 microns. So we're able to collect carrot seed crop debris with different particle sizes ranging from less than 50 microns to greater than 400 microns. This figure is showing the amounts of XHC in the different plumes that we sampled during carrot seed harvest. As you can see, we conducted 3 different experiments in 3 different harvesting events and collected anywhere between 4 to 8 samples per event. As you can see, XHC was present in all of the plumes that we sampled, indicating that the pathogen can persist or be present in these debris plumes that become airborne during carrot seed harvest. And in some cases, we found almost 1 times 10 to the 6th bacteria per gram of debris, or per filter, excuse me. We also saw effects of the distance from the plume, whereby the closer we were to the plume source, the more XHC we tended to capture. However, we were able to collect debris contaminated with XHC at distances up to 25 and 47 meters away, indicating that the pathogen can travel at least this far in airborne debris. On the right, we show the different filter sizes and the amounts of XHC that we collected in the different particle sizes. While most of the XHC that we recovered were in the higher size classes, 150 to 400 or greater microns in size, we did detect the pathogen in particles that were less than 50 microns in size, suggesting that these small particles could be an important source of long-range dispersal of the pathogen. We also conducted volumetric air sampling using Burkhardt spore samplers throughout the season and during the harvesting events. As you can see, in the summer of 2021, we were an average of 0.14 XHC genomes per liter of air were captured between May and September 7th. However, during the seed harvest event of that year, we collected over 10 times the amount of XHC compared to the season-long average, suggesting that these harvest events do contribute to airborne pathogen inoculum dispersal. Objective 4 involves the genome sequencing of a very diverse collection of carrot-associated Xanthomonas isolates. For this study, we sequenced the genomes of 312 strains of XHC and other carrot-associated Xanthomonas and bacteria. These isolates came from a diverse array of fields, seed lots, as well as air, soil, and symptomatic and non-symptomatic plants, and even plants other than carrot. These isolates were collected in multiple counties in Oregon, multiple states in the United States, and also included international isolates from various collections. And these isolates represented collection events dating back to 1957. In Dr. Jeff Chang's lab, he used whole genome sequencing using the Illumina HiSeq, MiniSeq, and Oxford Nanopore method, and compared all sequenced Xanthomonas from the NCBI database for context. Once sequenced, his lab conducted a multi-locus sequence analysis based on 6 housekeeping genes. Interestingly, all of the XHC strains that we included in the analysis belonged to a single lineage that were very closely related and had low genetic diversity. We also observed other Xanthomonas species associated with carrot, including Xanthomonas translucens, Xanthomonas campestris, Xanthomonas arboricola, and Xanthomonas vesicatoria. Although there was relatively little genetic diversity amongst our collection of XHC isolates from carrot, we were able to identify 3 major clades, and using an approach to time date them, we were able to determine that the first clade of XHC diverged around 1000 AD, soon after the domestication of carrots. There were 2 other divergence events, one in the 1600s and one in the 1800s. And there's a likely scenario that there were 2 or 3 primary introductions of XHC into agriculture early on, and then divergence and spread throughout the globe. Finally, for Objective 5, Dr. Gina Greenway has been working to develop enterprise budgets for carrot seed production in Southwest Idaho, Eastern Oregon, and Central Oregon. These enterprise budgets will be instrumental in helping us to establish and determine the economic impacts of bacterial blight in carrot seed production systems of the Pacific Northwest. So with that, I hope this presentation has given you a 30,000-foot overview of our research efforts related to this USDA NIFA SCRI project. Unfortunately, since I'm not here to answer any questions, please feel free to contact me either through email or with the phone number provided on the slide if you have any questions or comments about this project. Thank you again for the opportunity to share this work with everyone.
All right, so we do have a poll question though from Dr. Dung, so it should be up on your screen. We'll give this just a few seconds and then go over the correct answer. So true or false, burying crop debris is a potential management practice for bacteria blight in carrot seed crops. All right, so we'll share those results. And majority of you got it right. The answer is true.
All righty. Thanks, Julia. So now we have the time for our guest presentation. And I'm very pleased and thankful to Dr. Hartz and Kenneth Miller for accepting our invitation to present here at this meeting. And they will be presenting on a very interesting and hot topic, which is the nitrogen management and the groundwater protection process under the ILRC. Dr. Tim Hartz and Kenneth Miller.
Thank you. Okay. Yes, good morning, folks. My name is Tim Hartz. I worked for many years as an extension specialist at UC Davis, including with the carrot industry. And Jaspreet has asked us to do an update on the current status of the regulatory program that's underway in the Central Valley regarding the protection of groundwater from nitrate pollution. The first part of this presentation will be done by Ken Miller, who is a consultant with a company in Sacramento called Formation Environmental, who leads the industry effort to respond to this regulatory issue. Ken?
Thanks, Tim. Good morning, folks. Typically, the talk we give on the groundwater protection process runs about an hour, and I'm going to try to do it in about 10 to 15 minutes. So I will be going fairly briskly, and I'll try to hit on the main points. But if anyone has any questions or follow-up beyond what we can handle on the call, feel free to email me and we can try to get you some more information. But we'll sort of lay the framework on the GWP, groundwater protection, and then Tim can talk about specifically, managing nitrogen and carrot production sort of in this context of a regulatory program. Just real briefly, 13 of the 14 Central Valley Water Quality Coalitions are participating in this process. The only coalition not is Rice because they are not subject to the same orders, but all other coalitions have collectively come together to implement a unified approach for responding to the order. So what is the groundwater protection process? Essentially, there are three main components to the order. There's what's called a groundwater protection formula, which is essentially using data and methods to compute what is called the value. And what the value is a township leaching estimate. So that's a unified number, a total number rolled up to the township scale, represented only by irrigated agriculture. And then the target is basically where does that township need to get to in terms of loading to be protective of water quality, right, and achieve compliance. And so some townships may already be there. Some townships may require varying degrees of load reduction to meet those targets. And so in terms of the formula, and I'll talk a little bit more about this, essentially what we do in terms of data and methods are use grower reported data in the INMPs that you are all likely familiar with. And we basically integrate that into a process-based model to simulate leaching estimates. And that was approved by the regional board back in January of 21. We submitted the values using this formula in July of 21, and that was conditionally approved back in November of 21, and started the clock on trying to compute targets. And so targets were submitted this past summer. There was some discussion and back and forth, and basically identified some needed revisions. So we resubmitted just back in December of last year. And the targets themselves are still in review by the Central Valley Water Board, and so they're not yet approved. But one key piece of information to understand is that they will need to be revised and updated on a 5-year basis per the order. So we've proposed targets. We're working through getting them accepted, but there is opportunity for updates to them as sort of our ability to estimate them improves. So where does this apply? Effectively, the vast majority of the Central Valley, all of these blue outlined townships, there are 399 of them, are subject to this order. And it's all based upon how much high vulnerability area exists within the township. And so that was another sort of regulatory requirement coalitions had to do under their groundwater assessment reports, was delineate these high vulnerability areas, which you can see in red in the map here. And so take home is this applies to, just about the entire Central Valley. So to assess the root zone and basically implement the formula, the coalitions and growers ultimately opted to use what is called the soil and water assessment tool and to adapt that to the Central Valley, hence the name CV SWAT. Won't spend a whole lot of time going into the accolades, but it's been around a long time, developed by researchers over at USDA ARS and at Temple, Texas. It's heavily used by the scientific community, published widely. It's a publicly available model. You can go download it today for free if you'd like. And there are a whole list of reasons why we, and the coalitions opted to use this tool. And basically, it allows us to integrate a lot of spatial and temporal detail that we know matter when it comes down to trying to estimate what's going on in a root zone and what's leaving a root zone, right? And so SWOT allows us to pretty readily integrate sort of climate, soil information, detailed information. We provide detailed management information. So working with Dr. Tim Hartz about how to model carrots, for instance, in terms of representative management practices. It has an actual growing crop. So it's taking up nitrogen and water as a function of its growth stage and so on and so forth. And it's simulating, both the water and the nitrogen cycle in the root zone. So we could talk much more about this, but essentially it's a comprehensive model that can be applied across the Central Valley scale, but still give us sort of subfield scale modeling ability. So checks a lot of boxes for us. And it was adapted for the Central Valley in part. This work started, I guess, back in 2017, 2018 or so. So a lot of the work was funded by a conservation innovation grant from the Natural Resources Conservation Service that really helped us get a leg up and get this tool up and running. It's been further refined to a point where we now have more than 50 crop models calibrated to Central Valley conditions. So covering more than 98% of the cropped acreage, and again sort of integrated based on all the best available information, crop dynamics and management based upon experts like Tim, for instance. So it's a robust tool that the coalitions have developed to assess themselves and the root zone for a whole number of different applications. So when we actually apply it to the formula and calculate values, there are effectively 3 steps, and I'll go through these pretty quickly. We pull together all the data we need. We develop what we call the root zone library. It's just a big database of SWOT runs. And then we use that database to calculate root zone values. And so the main piece I'll focus on right now with data is the INP data that growers report. This basically tells us the range of fertilizer rates and the range of yields that are occurring within the Central Valley. And that helps us constrain or understand how many different modeling scenarios we need to run in SWOT to have a comprehensive representation of what's going on. So we take all of these possible scenarios, we run it through an automated procedure, and essentially run every possible scenario under the sun across all crops, soils, climate, management combinations, and build a big database, right? And so the idea here is we'll do all the modeling up front and then use that for years to come to compute these values. So essentially, and I'll talk about step 3 a little more on the next slide, but once we have this library built, we can then take grower data, look up the appropriate library run, and compute these values, which is the leaching estimate. So here's just an example township. You can see the different colored polygons here represent different soil types, so effectively different modeling units. The black outlines are parcels for which we have INP reports. So if we were to take an example parcel that actually has 6 different soil types within it, the workflow for computing leaching for this parcel would essentially be to take an INP report, look at the fertilizer rate applied, and look at the yield achieved. And then we go into our big database that's going to have model runs already done for carrot on these different soil types at that N rate and at that yield, or very close to it. And so we go in and we pull the appropriate match for each soil, which will have a leaching estimate associated with it, right? So we basically take those 6 numbers, we acre weight average them to give one number for the parcel, and then we do that for every parcel in the township and roll it up to the township scale. And so when it comes to reporting, everything is done at a township scale, so there's no specificity in terms of individual parcel contributions. There's no breakdown by crop. There's nothing like that. It's rolled up to an aggregate number at the township scale, but that aggregate is being comprised from sort of a subfield calculation. And what you can see on the right-hand side are the actual values, the current leaching estimates for townships that contain a fair amount of carrot acreage. These values are not only specific to carrot, it's a function of all the cropping patterns and management in that township. But just wanted to give folks a flavor for what these current estimates are looking like in terms of the relative contribution of carrot. That varies, but typically, you know, there's not many townships exceeding, say, 10% of the crop acreage in carrot based on the INP
You might
data.
want to specify what the target is, concentration was.
Sure. Well, let me get to that. Exactly. So that's load. So that's what's leaving the root zone. The targets are how much leaching is, you know, allowable to make sure that irrigated ag is not causing or contributing. And so what we need to do is essentially take those values that we computed and connect them to all of the other sort of components of the broader system that are ultimately going to drive water quality, right? And so that's accounting for recharge from various sources. It's accounting for other end loads that are impacting the aquifer, accounting for any post-root zone processes where they may occur, such as denitrification, accounting for groundwater processes, flow, transport, mixing, for example. And then ultimately, you know, having sort of a measurement to, or a model estimate to assess whether or not we're in compliance. And so what we have opted to use is a tool called the non-point source assessment tool developed by UC, and I have a slide on that. But ultimately, we're looking at produced water quality coming out of wells, because that's actually the water that is being applied to a beneficial use like drinking or irrigation. So what is NPSAT? I could spend half a day on it, and I will not now. Essentially, again, it was a model developed by UC researchers with state and federal funding. And so what that means is basically the coalitions were able to use sort of the state-of-the-art tool at very little cost to them for setting these targets, which was really a great partnership and collaboration with the university. It's based upon DWR's groundwater model, C2VSIM. And so that all the water budget information in terms of recharge from all the various sources is integrated into NPSAT. This is a model that's available to GSAs under the Sigma process. And so it's sort of the best available water balance information we could possibly use at this time. What it does is we're focusing on domestic wells. They are virtual domestic wells and not actual, only because they're not down to the exact XY location in the model. We don't know exactly where they are. We just know within sort of a 1 mile accuracy, but they're all based upon DWR's well completion reports. And so essentially what NPSAT does is it connects what's going on the surface, transports that to virtual wells. And then over time, we can assess water quality as a function of what's going on at the surface. Couple considerations: it's not accurate at the individual well level. You have to look across a big area. You'll never be accurate at a single well. So the minimum scale would be a township, which is sort of the scale we've applied it at. But it also gives us the ability to look outwards far into the future under sort of certain set of conditions to see what potentially will happen to groundwater if changes on the land surface today occur. So what are we trying to do? We're trying to leverage the best tools and information we have at the time. Again, targets will be revised on a 5-year basis. We're very transparent in our reporting that there is uncertainty in some key inputs into this framework that are going to affect the accuracy of our targets. So we know that we need to refine them over time. And we anticipate that under other regulatory programs like Sigma or CV salts, we should get some better information or refined information with respect to some of these key pieces here. And so it is very much an ongoing process. In terms of what we submitted to the regional board, there are 2 key pieces here. There are what are called interim milestones, interim performance goals that are milestones. And these are effectively sort of root zone based milestones that are intended to shift the industry towards meeting their targets. And Tim will actually kind of talk a little bit more about more or less the methodology that was used to develop them. But the whole concept here is to prioritize sort of the least efficient areas, work with them on outreach and education, and try to get them back to sort of efficiency levels that the vast majority of the industry is demonstrated as, doable. So that's the idea with the milestones. It's an interim performance goal. And then, of course, we've also submitted the targets themselves, which are based on groundwater modeling and are intended to protect water quality. Just a quick image showing a comparison. Not all townships have milestones. Some townships don't necessarily need them. The target is near enough or they're already there such that a milestone is not really warranted. But for those townships where there's sort of a greater distance to go, a milestone was supplied. And so you can see here just a general, possibility in terms of potential load reduction if across crops, management was, management implementation was, protective management practices were implemented to a greater degree. We could anticipate some sort of load reduction without substantial challenges or restrictions to the growers' operations. The other key piece is the targets themselves. Obviously more difficult than the targets, than the milestones. Again, they will be refined over time as more data and information are provided, and these are still under review at this point, so they're not yet accepted by the regional. What does all this mean? Some key points are the milestones and the targets are not enforceable metrics, so they're not limits. There's a regulatory difference there. That doesn't mean that in the future, the State Water Board may, consider what sort of enforceable metrics might be appropriate. So at this point, none of those exist. So it's really, if there's progress towards meeting milestones, towards meeting targets, that's sort of the best case scenario for the industry to, prevent things like these enforceable metrics from ever being created. So how do these get integrated into sort of the coalition's, framework? Coalitions are required to develop what are called groundwater quality management plans. And so these plans include schedules and milestones and performance goals, which will be the milestones and the targets that I had just talked about. And so if and when these targets get approved by the Water Board, they will be incorporated into the GQMPs, at which time the coalitions will sort of individually identify their outreach and education plans to work with growers to help understand this whole picture more and work towards actually meeting those milestones and targets. So I know that's a lot. That's an hour's worth of information in 10 minutes. Again, please let me know if you have more questions, but I'd like to leave some time for Tim to talk about carrots more specifically.
Okay, that's a good background to understand that where we are today with carrot production is that the estimated loss of nitrate to groundwater is still at a level of concern to the regulators, and some improved performance in terms of nitrogen efficiency will be required in future years. So that's where we begin. You've probably heard the expression A-R in this context of nitrogen management. Basically, it is just looking at the bald facts that you've got a certain amount of application. And in terms of these IMP reports that Ken has talked about, this is the total of fertilizer N, the available N in any organic amendments that you use, and the nitrate concentration or content of your irrigation water, total nitrogen that's going on that land surface. At the moment, our estimate is that the average amount of nitrogen removed from the field with a ton of carrots is 2.8 pounds. We'll talk about where that came from in a minute. And the end balance is this calculation, applied minus removed, or A-R. And the straight up fact that we can't ignore is the higher that balance, the greater risk to groundwater pollution with nitrate. To the point of where do we find that 2.8 pounds of N per ton of carrots number? My former colleague Daniel Geisler at UC Davis worked with industry groups to do observations of nitrogen content of harvested material. Carrot people worked hand in hand here the last couple of years, a total of 64 individual samples from commercial fields. You can see that there's a wide range of nitrogen contents, as you would expect, depending on yield and the amount of nitrogen applied. That 2.8 pounds per ton is just the average figure, so that's what we'll use for this presentation. If we look at that INMP data set, these are now grower reported nitrogen application and yield outcome reports. We've had a number of years now of these. I've graphed here the 2017 reported data and the 2020 reported data, 20 being the last full year that's now compiled. What you see is that the bulk of the acreage represented by these reports suggests that the industry is mostly using somewhere between 100 pounds to 200 pounds of applied. We have some growers less than 100. We have, unfortunately, as many as 30% of the acres at a application value above the 200. If you look at those that 4-year interval, what we see is there's very little difference, there's no obvious trend toward improvement of nitrogen use efficiency with carrots. It's basically people are doing what they have always done. So to this point, there's no implication that industry is responding to this challenge of improving nitrogen efficiency. I'm not suggesting that carrot is different in this regard. We see the same pattern with many other crops. To the point of a prior speaker, Ali from the Imperial Valley, what we see is that there really isn't a pattern to suggest that the higher nitrogen rates used by some growers are resulting in higher yields. These are the scatter diagrams of the grower reported data for these 2 years. Again, there's just no statistical observation of increasing N applied is increasing yield achieved. This again is not specific to carrots. There are a number of crops, mostly annuals, where we observe exactly this pattern. So what that gives us by default is if you're not achieving better yields with higher nitrogen rates, you are having a higher end balance with increasing nitrogen rates. It's just a fact that we can't ignore. Where does the current INP data put carrots in terms of ranking with other crops? It is a higher end balance than most perennials. In terms of other vegetable or row crops, it's kind of in the middle of the pack, but still at a level that is problematic for achieving the targets that Ken has talked about. Now, before anyone has a cow here, let's specify that the end balance is not exactly equal to nitrogen leach. We know that there are other end pathways that evolve, gaseous losses and potentially storage of nitrogen in organic matter in the soil. The reality, though, is that these processes probably account for no more than 10% of applied N. And for growers using a high application rate, typically that alternate pathway loss is not enough to bring them anywhere close to compliance. Rotational effects absolutely could be significant if behind the carrot crop you were to put in lightly fertilized wheat, for example. The net result across years would be a lower end balance. In reality, where carrots are grown, in a lot of cases, carrots will be rotated with other relatively highly fertilized vegetable crops. So this sequestration by rotation is not likely to be a huge factor in most cases. And it is also true that there are processes that Ken alluded to in the vadose zone, which is that zone below the root zone, but above groundwater, that might take a significant fraction of this nitrogen moved into that vadose out of the equation. But at the moment, that is something that's being actively trying to quantify. And at this point, we can't assume that we'll get a get out of jail free card because of these processes going on in the vadose. So where does that leave us? If you graph the 2020 INP data by the percent of total acres represented, what you find is that the median value of the A-R for carrots is 86 pounds of N per acre, meaning there's 86 pounds more applied than removed in reported yields. If you then look at the upper fraction of the acreage, the fraction where high rates of nitrogen are used, what you see is you start getting very, very high A-R values from those acres using high nitrogen application rates. And this is really where the focus has to be. The interim targets that Ken talked about are really focused at this 75 percentile level for carrots, and people over that level will be targeted for education activities, hopefully to help them bring down the amount of fertilizer they're using and or improve yields such that their A-R becomes lower. I would specify here that those high levels, 75th percentile up, are almost exclusively from rates above 200 pounds, which, agronomically are generally unsupported. So how might we improve efficiency? As Ali mentioned, irrigation efficiency is a big factor. Recognize, however, that there are limits on efficiency when sprinkler irrigation must be used. But even there's been enough work in the Central Valley to suggest that improving distribution uniformity of sprinklers and better scheduling can give us less leaching during the crop, and that will equate to less nitrogen movement or nitrate movement below the root zone. Another issue, and we've fought this battle on the coast for vegetable growers over there. There is a great reluctance on the part of growers to really credit the nitrogen involved in the irrigation, applied in irrigation water itself, the nitrate content of the irrigation water, and to think that that's equivalent to applied fertilizer. We did a whole range of field trials in commercial farms on the coast a few years ago, represented in this California Agricultural article, where we looked at that point exactly how efficiently could the nitrate that's in irrigation water be taken up. And the answer was, with appropriate irrigation management, a very high efficiency can be achieved, meaning that you can be comfortable using the nitrate content in your irrigation water in lieu of applied fertilizer. So if you look at the fact that a foot of water would carry a substantial amount of nitrogen in wells where you have reasonably high concentrations, surface water, for example, is typically 1 or 2 ppm nitrate N, which is negligible, and more or less forget that. But in wells in the San Joaquin Valley, there are some that go up to 20 or 25 ppm. And those wells have a substantial amount of nitrate, assuming that virtually every carrot field in the valley has at least a foot of transpired water by the crop. You can see that this could account for a substantial amount of nitrogen that you might be able to back off from fertilizer rates. I would be remiss if I didn't talk a little bit about organic production, because for carrots now, this is becoming a significant fraction of the industry. A point that is very clear in all the work that we've done in organic systems is that if you're going to be successful with organics, you have to enter a production season with a reasonable bank of soil nitrate in the soil, because you will have the crop will be taking up nitrogen faster than your soil itself can supply. So you have to begin with a bank balance, if you will, or you'll be deficient. So we observed in Central Valley organic systems, and this data comes from a study with processed tomatoes, but it's very applicable to carrots. The way that organics are managed with the amendments that are being used, what you end up with is many fields begin a crop with high nitrate concentrations. These data are for the top 2 feet of soil. So if you multiply the, say, 20 parts per million by 8 would be the amount of nitrate contained in an acre of ground. You can see that there are 150, even 200 pounds of nitrate-formed nitrogen as you begin a crop. That's easily moved beyond the reach of the carrots if your irrigation practice is not pretty good, and especially as you're germinating a carrot crop from seed. Leaching is inevitable, but the amount of that leaching needs to be minimized, or else you're going to lose a lot of your hard gained nitrate out of the system and possibly even end up on the nitrogen with the nitrogen short crop. So irrigation management early season in organic carrots is crucial for efficiency. Oops, I should have said that. Okay, so that's a long slog and we'll stop at that point. Only to observe that not only carrots, but other crops in the valley will be required to tighten up their nitrogen efficiency practices to come anywhere close to meeting these mandated groundwater nitrate targets. The way to do that, of course, is to start with the folks who are least efficient and work backwards, but we can't ignore the fact that even efficient practices today will have to tighten up to get to those target values. So I'll stop at that point. If we have questions, we'll take them.
All right, great. We do have one poll question for you guys, so we could get that launched up real quick and have you guys read that out for us.
How much nitrogen is contained in a ton of carrots on average?
Great. So we'll let people answer that question. And in the meantime, we do have a couple questions in the Q&A. So I can read those out to you guys. The first is, how do you distinguish between nitrate in groundwater caused by current conditions and that which is associated with the past? And how quickly do you foresee groundwater levels changing after current practices are changed?
Yeah, it's a really good question. And so going back to that groundwater model we used, NPSAT, it's very efficient to run it and to run it for long amounts of time into the future. And so basically what we do is more or less take current loading estimates based on the values and the swap model and all that. We take that and we take current hydrology and we run it out into the future. And so we're not really trying to assign, understand what happened in the past, what explains water quality today. We're trying to answer what will future water quality be under today's practices, right? So we don't get bogged down in that. We're trying to just look forward. So we kind of get around that issue.
Exactly.
All right, great. And then we have one more question here. It's kind of a long one. In your grower reported nitrogen application rates, does that include organic growers? And if so, how is the nitrogen from compost and organic fertilizers being calculated? Is that total nitrogen or the nitrogen that is expected to be available during the season from organic fertilizer addition?
Yeah, I will say that is one of the challenges in interpreting that data, because it's not clear whether any given report is organic or conventional. The instructions suggest that on the organic side, the number of pounds applied that you list should be your estimate of the available nitrogen from those amendments in that year. So if you were to apply 200 pounds of N as compost, you would have to estimate the fraction of those 200 pounds that would become available this year, and that is the number that you would use. it's very clear to me, having gone through these data for the past couple of years, there's still a lot of grower training that may be needed for clarity, because some of what we observe, both low values and high, I believe represent people who have not interpreted the instructions correctly. But overall, as I say, I'm close enough to the industry to know that application of 200+ pounds in conventional systems still is a relatively common thing, and that's the target zone that we're going to have to work with.
All right, great, thank you. And then we have the results of that poll up. So it looks like majority said 2.8 pounds. Is that correct? Yeah.
Yes, that is correct.
Perfect.
Yes.
All
Good
right,
job.
great. Good job, everybody. So with that, I'll turn it over to Jaspreet to round us out.
Thank you, Ken. Thanks, Dr. Hart. It was really nice for you to taking the time out today to speak at this meeting. And that brings us to the end of this meeting. And I would like to thank all of you for joining us today. And a special thanks to our speakers for presenting wonderful presentations. And that's all that we had today. Julia, do you have any announcements before we wrap up?
Yeah, just one last comment, just as a reminder, if you need California GPR credits, there's 1.5 hours of other today. You'll get an email by 5 p.m. today from A&R program support at ucanr.edu with a link to the final exam with those poll questions that we went through today on it, and make sure you take that within a week and pass with a score of 70% or better, and then you'll get your credits. So watch for that email if you need credit. Other than that, we're set to be done for the day.
All right, thank you again. And with that, I'll say have a good rest of your day, and thanks once again.